Hi students, welcome to Protein Synthesis and Gene Expression. You'll find it in chapter 9 of your Oxford textbook. So today we're looking at genomes and genes, the steps in protein synthesis and translation of the mRNA to an amino acid sequence to proteins at the ribosomes in the cytoplasms. This is a really complex process, but once you understand the steps, it will all come together and you will hopefully be understanding at a really high level by the end of it. So why are we looking at it? Firstly, down here, why are we looking at protein synthesis? Well, protein synthesis and polypeptide synthesis link to so many other parts of understanding within the syllabus and overall your understanding of biology. It's like unveiling the secrets of everything. It's using DNA to make physical things out of it. And it is the process of making you and who you are, all of your structures in your body. And once you learn about this, so many other pieces of your learning in biology over your high school years will come together. So just a little recap about some important vocabulary. So polypeptides are a chain of amino acids all joined together. Now we have 20 amino acids in the human body, and they are tiny spheres that act as the building blocks to everything in our body. So when you have many, many amino acids linked together, that will equate to a polypeptide chain. And the polypeptide chain can itself be called a protein once it has folded into a particular shape, or it can join together with many different polypeptide chains to form a protein. And this whole process is called protein synthesis. And a little recap on genome and genes, the difference between them. Well, gene is a section of DNA that carries the instructions for production of a protein, and the genome is the complete set of nucleotide sequences encoded in the total DNA of an organism. Okay, so those nucleotide sequences, the A, C, and the G, and the A, and the U, and the T, they're those nucleotide sequences. So the gene is just a section of those, and they are different lengths depending on what they are coding for. So the way I've structured this presentation, given the complexity of this process, is I've just provided a synopsis. So over the next 4 slides, I've provided a generalized overview so that you can see beginning to end what happens. And then after that, there's more detail in the slides about that process. I'm just trying to introduce you to this process using regular vocabulary so that you can understand simplified processes and engage with it, and then you can flesh it out as that vocabulary becomes more familiar to you. Okay, so this is an overview of transcription and translation and how protein synthesis occurs. So what you have here, I'm just going to grab a pen. So what you have here obviously is the cell, okay, and here is your nucleus, right? And this is where we're going to start. We're starting in the nucleus of your cell, and most cells in your body are just like this. So the DNA is here. But however, here's the catch. We cannot make anything in the nucleus. Everything that gets made in the cell gets made outside of the nucleus. So in this area of the cell, in the cytoplasm, where all those fantastic organelles are that you learned in grade 8 science or grade 9 science. So we need to get a message to the outside. So that's what this process is doing. So in the beginning, we call this transcription. So we're going to be transcribing down or transcribing down the information from the DNA so that we can take it outside of the nucleus and give it to a ribosome. So transcription starts with an enzyme called Rna Polymerase, which I think you've heard of before. And this enzyme attaches to the DNA strand and does pretty much 2 jobs at once. It unzips the strand and it breaks those hydrogen bonds. And as it is breaking those bonds, it then moves down the DNA. So where's it moving? Well, that all depends on what your body or your cell needs to make. So your DNA contains genes for everything that you make inside you, from muscles to proteins like insulin to hemoglobin, everything. So whatever it is that the body needs or what we need is where we start. So the RNA, so the RNA polymerase will attach to your DNA somewhere. It'll find a little start codon, and we'll look at codons in a second. And it will start to unzip it And at the same time it is unzipping it, it will start to make a copy of that code. And this is a complementary copy. So it's not going to be on a string of DNA that it is copying it to. It's going to be on a different strand called RNA, ribonucleic acid. And this RNA has a few different individual factors that make it unique. And we've looked at those in previous weeks. First of all, it's a single strand. And secondly, it has a different base from DNA. So instead of having thymine, it replaces this with uracil. That's very important to remember, as quite often you might have to transfer DNA to RNA and RNA to something called tRNA or transfer RNA, which we'll talk about shortly. And sometimes you might need to work backwards. So understanding these differences between DNA and RNA is very important. So the RNA polymerase, and we're still here in the DNA in the nucleus with the DNA in the nucleus. So imagine that the RNA polymerase is attached to our DNA somewhere, and it's moving down the length of the DNA. It's occurring here, and making a copy and transcribing it onto mRNA. Now, mRNA stands for messenger RNA. You're getting a message from your original DNA, making a complementary copy and sending that message outside of the nucleus into the cell. Okay, so once that RNA has been transcribed, as in we have that complete message to make whatever it is we're making. Just briefly, I will mention that before the RNA leaves the nucleus, it is processed slightly. So you'll have some of the bases that go into the RNA are not actually needed. So around about here, before it becomes that mature RNA and leaves the nucleus, they get some parts of that complementary copy will get spliced out or cut out and recombined. And the cut out parts are called introns, and the bits that are remaining, they're called exons. And the enzyme that does the splicing is called a spliceosome. It's a pretty cool name. Now, once that process has occurred, then the RNA strand can now be called mature RNA, and it's ready. We now have a complete message that has all the info we need to make whatever it is that we're making, and it's able to leave the nucleus and go into the cytoplasm. And when it enters the cytoplasm, it will be attached to one of the millions of ribosomes that we have around our nucleus. And these ribosomes are like the factories of our body. So imagine that this weird shape here is the ribosome. And they're going to be taking that message and turning it into real things. And this is where the magic happens. So once that mRNA, that messenger RNA, has attached to the ribosome, it can then be literally drawn through on it like a conveyor belt. And and it works in a very unique way. Now, importantly, as that messenger RNA goes through the ribosome can only read 3 bases at a time. And we call these 3 bases a codon. And it could be something like AUG. Now there's 3 bases, that's a codon. Or GUC, 3 bases, that's a codon. And now this is very important, and this will link to skills questions that might be often on an external exam. So these codons, whatever letter combination they may be, are always in 3s. And the reason they're in 3s is because they need to match something called an anticodon. And an anticodon will be up here. We'll talk about that in a second. So you've got your messenger RNA, and it's being the codon, those 3 bases at a time are being read by the ribosome. The ribosome is going to wait for another molecule to come over, and that molecule is called a tRNA transfer RNA. And this weird shape here, this is your transfer RNA, and at the bottom of it is an anticodon. So the tRNA are carrying these, as well, you see just above here, these little dots on transfer RNA, they're actually amino acids, the ones I spoke about at the start, that are like tiny spheres, and they are the building blocks to everything. So the tRNA, well, on the bottom of their structure, as I said, they have an anticodon, and these 3 bases will match exactly with the codon that's currently on the mRNA strand within the ribosome. So the ribosome will then bring these 2 together, and you can see that's depicted here. And when they have matched, the amino acid will be separated from the tRNA, from the transfer RNA, and will be left at the top of the ribosome. And this will happen again and again and again. And the mRNA will continue to be pulled through 3 bases at a time, and a matching tRNA molecule will combine and deposit its amino acid. And as you can see, those amino acids will start to join through peptide bonds. And once we have 1, 2, 3, 4, 5 amino acids all joined together, we can start to see that a chain is forming. This is called a polypeptide chain. Poly just means many. You have the words like mono, meaning one, poly means many. So this is a polypeptide chain. Now, once all of the amino acids have been deposited to make the necessary polypeptide, the process will stop. There'll be a stop codon, and the polypeptide will either form into that protein, it'll change shape and form into a protein, or fold and change shape, or combine with many different polypeptides to make a particular structure for whatever the body needs. That entire process going through that ribosome is known as translation. So we're translating that message that we took from the DNA, turned it into RNA or messenger RNA, put it into the ribosome. We are translating that message into something that is real. So this beautiful visual here is also depicted in this flow chart here. Following the arrows, DNA, transcription occurs, messenger RNA, that modification where those introns and exons are affected there. The introns are cut out and the exons remain behind. So then mature RNA is now there. Then it goes outside of the nucleus into the cytoplasm of the cell. Translation occurs, and we have proteins. A set of proteins expressed in a cell determine its phenotype. Remembering what phenotype means, what we physically can see of an organism, such as your hair color, your height, eye color, things like that. So that was huge. So I've written all of those processes down on 2 slides, slides 8 and slide 9. You can read through that again at your leisure. It really breaks it down simply to see from the start to the beginning, starting in the nucleus, the DNA is here, but we can't make anything. So that whole fabulous process of transcription occurs, essentially transcribing that information so we can take it outside of the nucleus and give it to the ribosome. Transcription starts with RNA polymerase. And before it exits the nucleus, some of those bases get spliced out, and then now it's considered mature RNA. And this is just part 2. Once it leaves the nucleus and is in the cytoplasm, I've just done a quick summary or a synopsis of everything I've just said, looking at that previous image. Now, as you do learn about gene expression, you will see a lot of different images, and I think it's very important to observe different images and see how they're depicted so that you're able to identify transfer RNA, the original mRNA strand, the codons and the anticodons, the the amino acids, what this is, the polypeptide chain, and so on. I have a great little video that I will show you now while we're talking that kind of animates this whole process.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called Rna Polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Let's look a little closer at those awesome shaped molecules, tRNA, transfer RNA, and how those anticodons are associated with that molecule. So the anticodon is found on one end of the tRNA and is matched to the amino acid found on the other end. And we've actually developed a great way to know or predict what that amino acid is going to be if we know the anticodon, which will give us an idea of what the codon is on the mRNA strand. I know that sounds a bit convoluted, but we'll jump in. So the anticodon is the complementary base sequence of that which is the code for that amino acid. For example, if the amino acid is holding on to methionine, then the anticodon will read UAC, and this will then match to AUG, which is the codon for methionine. Now, that doesn't sound like much, but what you will understand it when you learn how to read these codon charts, and let's have a look now. So I'll just move this over. So you might recognize that, okay, from a more simplistic view of a tRNA molecule. And you have in the brown, that's the anticodon, and in the green, this is a part of the codon from the mRNA, the messenger RNA strand. And this amino acid, which we don't know what amino acid it is yet, but we will once we use the codon chart and see what UUC is associates with which amino acid. So as I said earlier, there are 20 amino acids. So for example, if we had a codon, actually we'll use this one, UUC. So write down on your paper what UUC, and make sure you know that that is from the mRNA strand. So when we're trying to find out what this amino acid is, we do not use the anticodon on the tRNA, we use the codon on the mRNA. So let's move this over. Now on your codon chart, you have 3 sides, first base, second base, and third base. Now, knowing from earlier, let's go back again, UUC, you've got it written down. You're looking at this codon chart, you find U. Well, U is right here. We go the first base first. Let's grab a pen. And you know that that first letter will be in, we isolate this one, U. And the next one was U again. So we go to the second base and we isolate this one. Okay. So we know that in this square is going to be our amino acid. And remember, our third letter from the mRNA codon was C. So if we look over now at the third base, we see the C is right here. And if we go all the way along here, we can have a look and we know that the amino acid attached to that tRNA will be phenylalanine. Now, I want you to pause the video and try to find the associated amino acid for this codon here and then this codon here. So you should work with one first. So pause the video now. Let's take a look. So our codon, which would have been on the mRNA strand, mRNA, messenger RNA is AUG. So let's go to first base. We can see A. We know that we can isolate this row here. Okay, this is the long way to do it. We're going to do it the long way until you get it. The next is U. So you go to second base and you find U. And you isolate this one. And the third one is G. You go over to the third base. You know that it's over here in this square here where U and A combine. You find G, and over on the bottom, it's methionine. And that's actually a start codon as well. So let's look at the third one, AGU. So we go to the first base, A. You can isolate that row. Oh, I'm getting bad at that. G is over here, so we go to second base, the second level, and we isolate. We know already that it's going to be in this square here. Okay, and then our last one is U. So U is here, and we know, let me just clear that screen, it's called serine, or serine, this one here. So the amino acid that will be on the tRNA that is complementary to the mRNA codon AGU is serine. So now hopefully your vocab is a bit stronger than it was at the start of the lesson, and we're going to actually put this into practice, a bit of a protein synthesis race where you'll be going through the entire process as we've been talking about, and we'll do this in class together. So if you open up the presentation, which is 7 gene expression on Teams, you'll see on this slide there is a link underneath that will take you straight there, and I'd like you to start a new game. So as mentioned before, moving on with the same process, protein synthesis. So the following slides, which are numbered 16 to 34, provide additional details within which this process happens. Now you have an option to either go through these from 16 to 34, or you can head to Edrollo and view the associated video. It's a 27-minute video, and they have a lot of great questions embedded throughout. You can do either one of those, or you can actually do both. Now, there's a great video on protein synthesis for our more visual learners, and you're welcome to access that too. It's really good. And they also have a video about how to read a codon chart. There are actually 2 types of codons, a circular one as well. We're most likely using this one, so I didn't worry about showing you that one, but it does give you a really simple way of doing it. It does break it into 4, and I'm happy to help you with that in class as well if you need. But for now, decide which one you want to go to, and we'll work through that. I've just made this a bit shorter, so as you go through the rest of these slides from 16 all the way down, you'll have a lot more in-depth instruction with that, with protein synthesis. And yeah, please let me know if you have any questions, and if you missed a lesson today, hopefully this was helpful. You'll see that we've gone through the first 3 dots, and I'll create another video looking at the process, how gene expression can be regulated, looking at things like gene expression, acetylation and methylation, and we'll do a hands-on activity for this one. But for today, we looked at the first 3 pods here. Thanks, guys.